let's maybe hearing it from the expert's sort of perspective. What first even got you interested in this specific, I would say, technology application for early cancer detection?
Yeah, I think there's this tired trope that I think science is less about eureka moments and more about like, oh, that's interesting. Right? So the bottom line is we did not set out to solve like a liquid biopsy problem. We did not set out to figure out a way to do cancer detection from blood. That was kind of a happy coincidence of like the science that we were doing in the lab. Largely around this, cancer emergent RNAs that you mentioned. And we were interested in, during my work as a postdoc, I'd come across some of these RNAs. And it's just that has just stayed with me. That again, that's interesting. That there are RNA molecules that I only see in the context of cancer, but not in healthy cells. And when I started my lab, one of the we built our lab on multiple ideas, kind of like risk managed, right? So some of the less risky, kind of we know this is going to work, bread and butter, and some ideas that we're going to take, give it a shot and see what we see. So this was certainly in that category of high risk, high reward kind of projects that we were thinking about at the time with my kind of founding postdoc, Dr. Lisa Fish. Lisa was a graduate student in Sohail's lab where I was a postdoc, so we actually started my lab together. So she gets a lot of credit for actually having helped me build the lab. And she was an RNA biologist, and we had worked together earlier on a lot of kind of how RNA and RNA binding proteins work in the context of cancer. So the first problem that we started to think through was how many of these cancer emergent RNAs are there even there? So it turned out like a lot. So we systematically annotated over many years a lot of these cancer emergent RNAs, and we made interesting observations. One is that there's no shortage of them. There's tens of thousands of these RNAs, and more importantly, they are not appearing randomly. There is a logic to their appearance that is tied to the state of the cell, which means that a lung cancer cell will have different cancer emergent RNAs, which, as you said, we call oncRNAs. And interesting enough, onc is not cancer; it's orphan non-coding RNAs. Orphan in the sense that they come out of nowhere, and so these oncRNAs are tied to kind of the state that the cell resides in. Kind of like the same way that gene expression reflects on the state of the cell, and a lot of other high throughput measurements of the cell will also reflect that. But what is different about oncRNAs is that we started to think about them as kind of digital kind of barcodes of cell identity, right? Gene expression is quantitative. It goes up and down. If I have more of this gene and less of that gene, then I'm a cancer cell or a lung cancer cell. Whereas in the case of oncRNAs, you can say if I see this oncRNA and not that oncRNA, I would be a lung cancer cell. But initially, we were really interested in how these cancer emergent RNAs may even play roles in tumor progression. They could actually evolve new functions in the context of cancer. And that was the basis of the project by and large. But quite serendipitously, one day we made the observation that some of these RNAs are actually secreted because there are small RNAs, and cells do secrete small RNAs. And we have been actually interested in that process as well. And we had a project in the lab focused on understanding RNA secretion and kind of the underlying grammar of that in terms of which RNAs are secreted and which are not and why. And it turns out some of these oncRNAs tap into the same program to find their way out of the cell. And once we knew this, then we knew that there was a cancer detection application to look at there. And if I want to kind of maybe expand a little bit on that front, you have a tiny tumor in the body, but it's not isolated, right? It's still connected to the rest of the body, and via blood goes to the tumor, and the tumor relies on it for its survival and growth. And like any other cell, it does release material into the tumor, into the blood. So in principle, if you can catch what the tumor is releasing in the blood, you should be able to tell that there's a tumor there. Now the catch is that, well, all the other cells in the body are also releasing material into the blood, so it's a needle in the haystack problem. So you need some sort of really cancer-specific signal to be able to latch onto. So the first time that we tried to solve this problem, for example, was with mutations. Like mutations in cancer, somatic mutations in cancer, are cancer-specific. So if you can find them in the blood, then it means that there's a tumor that's releasing them. It turned out it's a bit more complicated than that, and so mutations for early cancer detection were not enough of a kind of signal-to-noise ratio, but DNA methylation emerged kind of as kind of the right approach for reading a cancer-specific signal from the blood. So these are pieces of DNA that are differentially methylated only in the context of cancer, and then are released by cancer cells because cancer cells die and they release the DNA into the blood, and you can sequence the entirety of the DNA in the blood. Of course, there's DNA from the other cells, but you know exactly what patterns of methylation you're looking for, and if you find them, then it means that there's an underlying tumor that's releasing them.
Could you explain quickly what methylation means?
So methylation is basically, there is this gap between kind of DNA and RNA, kind of how is DNA regulated so that not all parts of DNA are turned into RNA, and specific parts are kind of transcribed and expressed in a specific cells. The idea is that every cell in our body shares the same genetic blueprint, but we get more than 200 cell types in our body. All of that is based on how every gene is expressed, where it's expressed, how much it's expressed. And so this is the type of regulation that sits on DNA that does that in every cell. And one way that that process is controlled is via marks on DNA itself. Which is this methylation marks. So methylation is a chemical group that sits on DNA. You can add it, you can remove it, and that can silence or activate a gene based on its proximity. But again, because a lot of dysregulations happens in the context of cancer, one of these dysregulations is emergence of these methylation patterns that are cancer-specific, the same way that there are these oncRNAs, there's RNA species that are cancer-emergent, the same that are mutations in DNA are cancer-emergent. So all of these are kind of cancer-emergent signals, but the question is how reliably can you actually detect them from the blood? And the challenge with DNA, on the other hand, as effective as it has been, is that cancers release very little DNA into the blood. So it's just it's very good signal, but it's very faint signal. And so it tends to kind of skew towards later stage cancers for you being able to detect them. Now, in case of RNA, and specifically oncRNAs, cells release them actively. So it's not kind of a dead cancer cell event, it's a live cancer cell. Cells, even normal cells, are actively releasing RNA, small RNA, as I mentioned, into the blood. So kind of you can have like orders of magnitude more signal. OncRNAs allows you to focus that signal on cancer cells and be able to know if I see these oncRNAs in the blood, I know that there was an underlying tumor that has released them. And we have built these patterns of oncRNA presence and absence that we are basically looking for in the blood.